Okay, let's start our discussion about or continue our discussion on the different kinds of linkages that we can see, right? So like you have these monomers and then these two monomers come together, something is released and they form a bond, okay? So we saw this kind of linkage in amino acids, in which you have one amino acid, so I hope by now you can understand what an amino acid is, and so this is the amino acid. Now what will happen is this OH and this NH2 will come together, these two ends will come together and they will release water, okay? So you end up minus water, so a water molecule comes. So these two things come together, a water molecule is released and you end up getting, and this could be, you could also look at this as the Zwitter ion, or maybe let's keep it simple, in which you have this OH, the water is released and you end up having this C, H, R. What will you get in between? What is the molecule that comes in here? Is it an O, an N, a C? What do you get here? What is this? What is this? Like N, right? So this is that N. This N, there is an H already attached to it. There is a CH, R dash, and the COOH. This linkage is called the peptide linkage, right? So this is what we saw. So this was a linkage. This particular type of linkage was called peptide linkage. So when you have all these different kinds of, so you can have multiple of these peptide linkages, so, and you can form a sequence of peptide linkages, and that sequence is called a polypeptide. So it's a polymer of the peptide linkages. So this is also called a polypeptide. Right, so like the primary sequence is actually called the primary sequence. You can then have secondary sequences. All these, you can imagine these different chains getting entangled and whatnot. So you can get secondary, tertiary, and so on and so forth, right? So these then become secondary, tertiary, and so on, and finally, so that those are the protein structures, right? So yeah, amino acid getting to a polypeptide, getting to primary sequence, secondary sequence, tertiary sequence, and the protein, right? So this was one type of the linkage. Let's move on to another type of a linkage. Okay, before we do that, tell me, what will you have if you have, so if you have this COOH group and then R dash. So what is this? What is a functional group? So this is a carboxylic acid, and what is this? Alcohol. So this is a carboxylic acid, this is an alcohol. If you think very similar to the polypeptide, what will happen when these two things come together? you know, like that NH2 came and the COH came, so that OH from the COH and H2, so all that thing came and the water was released. A similar thing will also happen in here. So what will that? What is that? Think. So go into your that sheet that I gave you. What is the, right? So in here, what will happen is, this water is released. So OH from the COH and H from the alcohol is released. So you get a minus H2O in here, and you end up getting an RCOO R dash. This is an ester. What will you call this process? You're producing an ester. What will you call that? So this will become esterification. Esterification. This is a very important reaction. I'll just give you a small example, but this is not the main topic, but I'll give you a small example because we are talking about this. So whenever you have, probably it's not that common in here, but in India we have some problems of and in many countries probably, why just call out one country. When I was growing up, we used to hear that people died after eating poisonous alcohol. Okay, so there is this thing. So this is related to that. So sometimes ethyl alcohol, which is CH3CH2OH, is alcohol that we can drink. That's what is sold. That is not poisonous in small dosages. I mean, eventually, large dosages, it affects your body. But methyl alcohol, CH3OH, is very dangerous. If your alcohol making process was not accurate and you ended up making methyl alcohol with the ethyl alcohol, then that alcohol becomes pyreated. So that's not good. When methyl alcohol enters your body, it gets converted to methyldehyde or methanol. That methanol gets converted to formic acid, right? That is very dangerous. Now, in order to remove that formic acid, what special technique has been utilized is this technique. So imagine that your body has a lot of, say, formic acid, and you want to remove this. What you actually do is drink some more alcohol. I mean, not drink it, but it is being medically administered in your body. But you get another alcohol. These two combine and your body secretes out ester. So like an example of that, right? Esterification. Now, this water thing you're probably getting used to now, right? Now imagine what will happen if you have two carbohydrates together. Now, first let's think about what is a carbohydrate. The name is very similar to hydrocarbons. But you can see there is a hydrate in here. So that is giving you some idea about a water or water related thing. So what this is an organic compound which has 4 to 5 carbon atoms in it. It has multiple OH groups in it, and at least one, so it has either an aldehyde on top of this OH, an aldehyde or a ketone. So it has multiple OH groups plus either an aldehyde or a ketone. That's what a carbohydrate is. And there could be different kinds of carbohydrates. You could have monosaccharides, you could have what? Disaccharides, in which you get two of these things coming together forming a polymer, okay, or a dimer, okay. You could have oligosaccharides, in which you get more than two, but not hundreds or thousands, okay, maybe within hundreds, okay, hundreds of molecules coming together and doing that. But then you can have polysaccharides, in which you can get thousands and thousands of these individual units or monosaccharides coming together and forming these polysaccharides. Okay, so let's look into one of these structures, and then we will try to understand how they form the dimers, because it will be very similar to that water removal. So these OH groups coming in, the water is being removed. Okay. Now there are different ways of representing the organic molecule, and especially the sugar. Okay, so I will draw the same molecule glucose. Okay, can anyone tell me the formula for glucose? Okay, now let's see whether this is correct or not. Now there are various ways of writing this. The simplest way that we already have seen, that stick model, is this. I will name these. So I'll start with this carbon, will get will become carbon number 1, because this is the aldehyde group. This has a higher precedence over the alcohols. Okay, so we'll name it 1, 2, 3, and then I'll go sequentially. So there are 6 carbons. Okay, now the same molecule can be represented also using this. So this is that first carbon, this is the second, third, fourth, fifth, and this is the 6th carbon. It's another way of representing it. Now, there is a very special thing that these sugars do when they are dissolved in water. Okay, what they will do is, now this OH is going to attack this carbon center. Why? Because this carbon center is what I call nucleophilic center. Why is it nucleophilic? Because this carbon, this double bond, sometimes gets broken, and you get, if that double bond is broken, because double bonds are easier to break, right? Because it's a pi bond, because it's not a direct overlap, it's an interaction, right? So it's easier to break. If this breaks, who is going to take the electrons? Oxygen or carbon? Oxygen, because it is more electronegative. So this becomes what? Positive, right? This becomes positive. Who will be attracted to a positive center? Who has a negative charge or an electron? Because opposite charges attract each other. So if somewhere there is a positive charge, I will get attracted to that if I have a negative charge. and a positive charge. Who has a positive charge? An electron or the nucleus? The nucleus typically has. So that's why we call it a nucleophile, because it has a positive charge. So it becomes a nucleophilic center. So anything that loves positive charges will come and attack. This oxygen is going to come here and attack this first carbon. Okay, and what you will get is this. So you get a 6-membered ring. Okay, now this carbon is the first carbon, the second, third, fourth, fifth, and on this fifth carbon you have a CH2OH group. So in here I have a CH2 and an OH group. On this first carbon, now you get, so this is that O, this was that O, right? This is that O. So this is the that O came here, and this minus charge was broken. This got a minus charge. Now this oxygen already had 2 bonds, one with this carbon and another with hydrogen. If this is attacking, it has to release one of its bonds, right? So this hydrogen was released and was taken up by this oxygen. Okay, that is why you get in here, you will get that OH. So this carbon 1, which was the aldehyde center or the carbonyl center, that's what so carbonyls are 2 types, aldehydes and ketones, both are called carbonyl compounds. So this was the nucleophilic center. So this carbon has now this OH, which is this ketone. This is the O, which is here. The ringed O is this, not this. Okay? Now same, you get the OH here, OH, OH. Okay, so the formula of even this is C6H12O6. This is also sugar, this is also glucose, but in water this is not. So if you have water, so there is an equilibrium that exists between that structure and this structure. Okay, so when you dissolve sugar, so if you have solid sugar in your bottle, which is this, dissolve in water, you get an equilibrium between these 2 things. Okay, there are many things to understand and know, but we are not trying to understand that entire chemistry, just the structure. Okay, so this is another way of representing it, and there is also a simpler way of representing it. Is let's stick to this. This is the more common way of representing. So you will see this a lot. Now there is a special thing that happens. Now we'll have to think about this group OH and this CH2OH group. Now this, think about it, that this structure can be represented in 2 ways or multiple ways, but let me represent it in another way. So when I'm showing OH down, so there is an H down, which I'm not showing. Okay. What is the difference between these 2 structures? two structures?
Position of that OH that is connected to carbon 1, that is changed.
How is it changed?
Mirror image.
Not exactly. This and this. So if you imagine this to be a planar structure, if you think like that, then this CH2OH group and this OH groups are on opposite sides or opposite sides of the plane. In this, they are on the same side. Okay, that's the difference. So we can call this as the trans form of the glucose, and this we can call what? The cis form.
Because the two groups
Because are on the opposite sides. This is also called the alpha form, the trans form, and this is called the beta form. Now, as you will also see that there could be, sugar is optically active. Okay, so you will also have you will have enantiomers, right? So you can have a D enantiomer of that sugar or L enantiomers of the sugar. Okay, dextro sugar or levo sugar, two types of sugars you will have. Now, life will generally prefer, in this case, the D sugars. I'm not getting into the enantiomeric things, I'm just letting you know that both DLN sugars are present, but life seems, at least life on Earth seems to include D sugars only in their making or in their business. What is different from the amino acids is this. Amino acids, so there are 20 essential amino acids for life, as we discussed in the last class. One of them is optically inactive. What's the name of that? Glycine, in which the R is an H, right? So the R form is an H. So it has two hydrogens, that alpha carbon has two hydrogens, and that's why it is not an optically active. So that's, but rest, there are 18 amino acids are used in their levo forms or the left-handed forms in life. Only one other is used in the dextro form. This is a little different than what life is doing for the sugars. It's sugar, we are not seeing the levo forms. We are seeing complete selectivity of D sugars in here, okay? Now let's try to see how two of these sugars will come together. Before that, so this was your glucose. Let me write a few of the other sugars. Let's write fructose. This is your fructose. Similar to that. So you can also write fructose in terms of this and this. Can someone look at this and say whether the fructose, what is the relationship between fructose and glucose? What are they? Very simplistically, what is the formula for fructose? It is the same. What do you call those things? Isomers. This is an isomer, right? The fructose form, what do you think is the carbonyl group? Aldehyde or ketone? This is the one carbon. I mean, this is the one, but yeah, this is where the attack is happening in the 2. Think about this. So this carbon, this oxygen attacked this carbon, which was at carbon number 2, correct? So if you try to draw this structure, so the carbon number 2 probably had the double O, that's why it attacked here. So if this happened at carbon number 2, what will this be? Because the carbon number 1, you can see this, so there is a CH2OH here. So whether will this become an aldehyde or a ketone?
Yeah, because it's double.
It will be a ketone. So imagine that this entire thing remains the same, except that this second carbon becomes the seat of attack. In here, and that's the difference between this and this. Because the attack happened on this carbon, which is a ketone, you ended up getting a 5-membered ring here. In here, you were getting a 6-membered ring. 6-membered rings are more stable because there is less strain on those. Because each of these carbons are what? sp3 hybridized. Correct? So they would want to have a 109 degree kind of thing, which is still possible in here. But as you close down the structures, 4-membered ring, 3-membered ring, they become more and more unstable. So when you increase from 3 to 4 to 5 to 6-membered rings, your stability increases. But then again, in 7-membered rings, there is a stability problem. So we have seen that, like in generally, we see that the 6-membered rings are the most stable. 5-membered are possible, but yeah, I mean, not as stable as the 6-membered. Okay, so the glucose and fructose are essentially the same, except that double bond O, that ketone group, I mean, the double bond O, the carbonyl group is coming at the second center. Okay. All right. I will also write another molecule called ribose. So these are 6-membered rings, so we also call them hexose. So what will you call the fructose? So that's a hexose sugar. What will you call the fructose? It's a pentose sugar. It's a 5-membered ring. Is the ring. It's called a pentose sugar. It's a 5-membered ring. We also see ribose, which is also a 5-membered ring. This is a ribose sugar. This is also called a pentose because it's a 5-membered ring. Okay, this will be involved in RNA. Because what is the full form of RNA? Ribonucleic acid. So this is the ribo part of the ribonucleic acid. And what is DNA? Deoxyribose. So you just remove this, and this becomes a deoxyribose group, and that's what gets attached to the DNA structure. Okay, now we kind of understand like the different structures. Now we'll see the similar kind of linkage in 2 glucose molecules, say for example. Okay,